Hello everyone, I am Dr. Trupti and you are watching my YouTube channel Enjoy Biochemistry. In today's video, let's learn about vitamin B12. At the end of this session, students shall be able to describe the sources, RDA, biochemical functions and deficiency manifestations of vitamin B12. Vitamin B12, it is the hematopoietic water-soluble B complex vitamin. This is required for the maturation of erythrocytes. And it is also called as extrinsic factor of Castle because this is the factor which is derived from the external sources. It has other name, that is anti-pernicious anemia factor, because its deficiency is associated with the condition called as pernicious anemia. This condition is caused due to deficiency of intrinsic factor of Castle, which is required for absorption of vitamin B12. All vitamin B12, they are called as cobalamins because it has cobalt. This is the structure of vitamin B12, that is cobalamin. It has tetrapyrrole ring, 4 corine rings and central cobalt atom. This central cobalt atom, it is linked with the 4 nitrogen atoms of corine rings and thus it forms 4 coordination bonds. The 5th bond is formed with the nucleotide side chain dimethyl benzimidazole, and the 6th bond is formed with the various functional groups like CN, methyl, hydroxyl, deoxyadenosyl, and depending upon the presence of the groups, it is called as cyanocobalamin, methylcobalamin, hydroxycobalamin or deoxyadenosyl cobalamin. And the important feature about vitamin B12 is that it is synthesized only by microorganisms and not by animals and plants. There are mainly 4 forms of cobalamin. The first one is methylcobalamin. Second is 5'-deoxyadenosyl cobalamin. Third is hydroxyl cobalamin. And fourth is cyanocobalamin. Both methylcobalamin and 5'-deoxyadenosyl cobalamin, these are the bioactive coenzyme forms of vitamin B12. And this deoxyadenosyl cobalamin, it is the predominant coenzyme form which is present in the mitochondria and it is the storage form. Mainly it is stored in the liver, that is vitamin B12 is stored in the form of deoxyadenosyl in the liver. Methylcobalamin, it is the coenzyme bioactive form and it is mainly present in the circulation, that is in the blood. It is transport form which is present in the serum as well as it is also present in the cytosol. It is the intracellular form of vitamin B12. Hydroxycobalamin, it is the natural form of cobalamin and it is the intracellular intermediate form and it is present in the injectable preparations. While cyanocobalamin, it is the synthetic cobalamin. It is present in the commercial oral form and this cyano group, it has no physiological function. So there are 4 forms: methylcobalamin, deoxyadenosyl cobalamin, hydroxycobalamin and cyanocobalamin. What are the various dietary sources of vitamin B12? We know that vitamin B12 is synthesized only by the microorganisms, so it is not derived from the plant sources. The various animal sources are liver, eggs, milk, oyster, chicken, pork, and curd is better source than milk because the lactobacillus in curd synthesizes vitamin B12. It is synthesized only by anaerobic microorganisms and animals obtain this B12 either by eating food derived from other animals or from the intestinal bacterial flora synthesis. It can also be derived from the fortified food, for example flour, ready to eat breakfast cereals and bread. Humans get very small amount of vitamin B12 from their intestinal flora and this vitamin B12 is a unique water soluble vitamin which can be stored in the body and the body stores means it is stored in the liver. It is stored in 4 to 5 milligram quantity which is enough for 3 to 5 years. That's why deficiency of vitamin B12 manifests very late. What is the recommended dietary allowance for vitamin B12? In adult male and female, the RDA is 1 to 2 microgram per day. In children, it is about 0.7 to 1.4 microgram per day. Its requirement increases in pregnancy. It is 2.6 microgram per day. And during the lactation, it is about 2.8 microgram per day. Therapeutic dose of vitamin B12 is 1 milligram per day orally. Vitamin B12 has highly specialized mechanism for its absorption, and it involves various vitamin B12 binding proteins like transcobalamin 1, intrinsic factor of Castle, and transcobalamin 2. This transcobalamin 1 was earlier called as R binder. It is also called as cobalophyllin and haptocorin. This is produced by salivary glands and parietal cells of stomach. What is its role? It protects the acid-sensitive vitamin B12 while it moves through the stomach. This intrinsic factor of Castle, it is 50 kilodalton glycoprotein which is secreted by the gastric parietal cells, and its secretion is stimulated by various mechanisms which are involved in the gastric acid, that is hydrochloric acid secretion, for example histamine, acetylcholine and gastrin. It stimulates the secretion of this intrinsic factor of Castle from gastric parietal cells. And this transcobalamin 2, it is involved in the transport of methylcobalamin in the circulation. Now let's see what happens in the stomach and duodenum when food of animal origin containing vitamin B12 is ingested. It is mixed with the haptocorin which is released or secreted by the salivary gland. This haptocorin is also called as cobalophyllin. So first, the food protein containing vitamin B12, it is mixed with cobalophyllin, and when it reaches to the stomach, there the gastric acid, that is hydrochloric acid and pepsin, it is responsible for this release of vitamin B12 from the food. So vitamin B12 is released from the food, and now this cobalophyllin, it binds to this released vitamin B12. So vitamin B12 now binds to the cobalophyllin, and then this complex of vitamin B12 and cobalophyllin, it reaches to the duodenum. Here in the duodenum, the cobalophyllin, it is digested by the pancreatic enzyme, that is trypsin. So now only vitamin B12 is here, that because the cobalophyllin, it is digested by trypsin. Vitamin B12 alone cannot be absorbed by the enterocytes, so it requires one important glycoprotein that is called as intrinsic factor of Castle, which is secreted by the parietal cells of stomach. So this intrinsic factor, it binds to this vitamin B12 in the duodenum. Then this intrinsic factor and vitamin B12 complex, it reaches to the terminal ileum where the absorption of this complex occurs in the enterocytes. This complex binds to the tubulin receptors present on the enterocytes, and then this complex is internalized. So this vitamin B12 requires intrinsic factor for internalization of vitamin B12 in the enterocytes, and this is the energy dependent and calcium dependent process. So once this complex is internalized, then the dissociation of complex occurs. So this intrinsic factor, it is dissociated from vitamin B12. Now this free vitamin B12, it is methylated to form methylcobalamin in the cytosol, and then this vitamin B12, it binds to the transporter, that is transcobalamin 2. In the enterocytes, this vitamin B12 and transcobalamin 2, this complex reaches to the basolateral membrane of the enterocytes, and here only free cobalamin, it is transported in the circulation, and here it binds to the transcobalamin 2, and then this transcobalamin 2 and B12 complex, then through the portal circulation, it reaches to liver, and in the liver it is stored in the form of adenosyl cobalamin. Here this transcobalamin is degraded, and free this deoxyadenosyl cobalamin, it is stored in the liver. Some amount of this complex, it is taken up by the other tissues. Here also free B12 is dissociated, and then this is utilized by the tissues. Now this vitamin B12, it has an extensive enterohepatic circulation. this vitamin B12 is transported from the liver via bile duct to the duodenum, and vitamin B12 is continuously secreted in the bile, but most is reabsorbed and is available for metabolic functions. And if the circulating vitamin B12 concentration exceeds the binding capacity of blood, then excess will be excreted in the urine. But in the most circumstances, the highest losses of vitamin B12 occurs through the feces. In the circulation, 70 to 90% of vitamin B12, it is bound to haptocorine, and it is called as holohaptocorine. It is metabolically inactive form. And 10 to 30% is only metabolically active form, which is bound to transcobalamin, which is also called as holotranscobalamin. What happens in the target cell? What is the metabolic role of vitamin B12? It is important in two biochemical reactions. So when from the blood this methylcobalamin is taken up by the target cell, it can be directly used by enzyme methionine synthase for regeneration of methionine from homocysteine. And if it is required in the second reaction, that is by the methylmalonyl-CoA mutase, then it is converted into hydroxyl cobalamin by tetrahydrofolate dependent reaction, and then it is sent in the mitochondria where it is converted into adenosyl cobalamin, and it is used for this enzyme methylmalonyl-CoA mutase, which is required for formation of succinyl-CoA from methylmalonyl-CoA. And these both forms, that is methylcobalamin and adenosyl cobalamin, these are interconvertible according to the cellular need. What are the various biochemical functions of vitamin B12? It is required for the effective hematopoiesis, DNA, RNA production, methionine and choline synthesis, then myelin sheath formation and regeneration of folic acid. Now let's see the metabolic role of first coenzyme of vitamin B12, that is methylcobalamin. It is required for the enzyme methionine synthase. Methionine is formed from homocysteine by the action of enzyme methionine synthase, which is also called as homocysteine methyltransferase. This reaction requires folate as well as vitamin B12, that is methylcobalamin. Now the methyl group is donated by this N5-methyl tetrahydrofolate to the cobalamin, and now this cobalamin becomes methylcobalamin, and N5-methyl tetrahydrofolate is converted to tetrahydrofolate, which is utilized in the 1-carbon pool. Now this methylcobalamin, from this methylcobalamin, methyl group is given to homocysteine, and now homocysteine becomes methionine. And then how this methyl group is further used in the transmethylation reaction? Methionine cannot donate its methyl group because it is not the active methyl donor. So it has to be converted into active methyl donor, that is S-adenosyl methionine, which is also called as SAM by the action of enzyme methionine adenosyl transferase, and this reaction requires ATP. Further, this S-adenosyl methionine donates or transfers its methyl group to the acceptor to form the methylated product. So this is involved in the transmethylation reaction, and this happens with the help of enzyme methyl transferase, and here S-adenosyl homocysteine is formed, which is further converted to homocysteine by adenosyl homocysteinase, and later on again this from this homocysteine methionine is regenerated. Now if this cobalamin is deficient, then the N-methyl tetrahydrofolate cannot transfer its methyl group to this cobalamin, and homocysteine cannot be converted to methionine, and this tetrahydrofolate, it is trapped in the form of N-methyl tetrahydrofolate in cobalamin deficiency, and it is called as folate trap because here due to vitamin B12 deficiency, folate deficiency also occurs. And as there is a cobalamin deficiency, methionine cannot be formed, and further S-adenosyl methionine cannot be formed, so the transmethylation process is also hampered. And this transmethylation or methylation, it is very important process which is required for the DNA methylation, which has role in controlling DNA as well as the epigenetics. It has role in repair and regeneration of DNA and RNA, various immune functions, detoxification, even it is required for the phospholipid methylation, and also synthesis and regulation of neurotransmitters and hormones. Now let's see the role of deoxyadenosine cobalamin. From oxidation of odd chain fatty acid as well as from the metabolism of amino acids like valine, isoleucine, methionine and threonine, propionyl CoA is formed. From this propionyl CoA, dimethylmalonyl CoA is formed by the carboxylation reaction. This reaction is catalyzed by propionyl CoA carboxylase and it requires ABC, that is ATP, biotin and carbon dioxide. Then this dimethylmalonyl CoA is converted to L-methylmalonyl CoA, which is further converted to succinyl CoA, and this is very important reaction where deoxyadenosine cobalamin is utilized, and this reaction is catalyzed by methylmalonyl CoA mutase. Further, this succinyl CoA, as it is the intermediate of TCA cycle, it will be utilized in the TCA cycle. Now, if there is deficiency of vitamin B12, then there will not be formation of succinyl CoA. So the level of methylmalonyl CoA in the blood is increased and further it is excreted through urine, and there will be the condition called as methylmalonic aciduria, that is excretion of methylmalonic acid in the urine. We have seen the metabolic role of vitamin B12. Now we'll see the causes of vitamin B12 deficiency. First is nutritional, and this vitamin B12 deficiency is common in vegans because they don't consume animal products as well as dairy products, and also it is common in malnutrition. And what are the various causes where the vitamin B12 absorption is reduced? So there are various gastric disorders. For example, achlorhydria. Pernicious anemia is also important cause where intrinsic factor is deficient, and it is the autoimmune disease which is due to the parietal antibodies against the parietal cells, and due to lack of intrinsic factor vitamin B12 cannot be absorbed. Gastrictomy, gastric atrophy, it leads to the deficiency of intrinsic factor, and further it is responsible for the decreased absorption of vitamin B12. Now what are the various intestinal disorders which are responsible for decreased absorption? So if there is ileal resection, because vitamin B12 absorption occurs in the terminal ileum in conditions like sprue, tuberculosis, even in the pancreatic diseases the absorption is retarded. Various drugs are also responsible for retarding the absorption like metformin, colchicine, and vitamin B12 deficiency is also common in fish tapeworm infection. The most common cause of vitamin B12 deficiency is associated with the pernicious anemia, which is due to deficiency of intrinsic factor. However, it is rare in India. What are the deficiency manifestations of vitamin B12? Due to deficiency of vitamin B12, folate trap occurs and it results in the folate deficiency, and it is manifested as megaloblastic anemia. So the cause of megaloblastic anemia is either folate deficiency or vitamin B12 deficiency or deficiency of both. Then as there is a deficiency of vitamin B12, homocysteine cannot be converted to methionine. So there is accumulation of homocysteine which results in the increased concentration of homocysteine in blood, and further it is excreted through urine resulting into homocystinuria. And increased homocysteine in the blood, it increases atherosclerosis and responsible for coronary artery disease. The mechanism is already described in the video on folic acid. And this vitamin B12, it is required for myelination, so its deficiency results in the demyelination, and it is also responsible for subacute combined degeneration. Various nail changes can also be seen in the deficiency of vitamin B12, that is parallel ridges on the nails are observed. The clinical manifestations of vitamin B12 deficiency includes the manifestation due to anemia, GI manifestation and neurological symptoms. Both folate and cobalamin deficiencies have similar anemia and gastrointestinal symptoms. So what are those anemia symptoms? These symptoms include fatigue, weakness, palpitation, vertigo, tachycardia. Gastrointestinal symptoms include sore, beefy red tongue, weight loss, diarrhea. So these symptoms are common in both folate and vitamin B12 deficiency, but neurological symptoms are exclusively seen in vitamin B12 deficiency, and these includes numbness, paresthesia, ataxia, abnormal reflexes and diminished vibratory sensations. What is the biochemical basis of megaloblastic anemia seen in vitamin B12 deficiency? We know that for regeneration of methionine from homocysteine, we require cobalamin as well as methyl tetrahydrofolate. Now, in this reaction, tetrahydrofolate is formed, and from this tetrahydrofolate, methylene tetrahydrofolate can be formed by the interconversion of serine to glycine. This methylene tetrahydrofolate can be converted to N5-methyl tetrahydrofolate, but this reaction is not reversible. This methylene tetrahydrofolate, it is converted to dihydrofolate, and in this reaction the deoxyuridine monophosphate is converted to TMP, that is thymine monophosphate, and this is required for the DNA synthesis. Then this DHF is converted into tetrahydrofolate. So this vitamin B12, it is required for recycling of tetrahydrofolate, and it is also carrier of methyl group because the methionine leads to formation of S-adenosyl methionine, which is the active methyl donor. Now what will happen if this cobalamin is deficient? Then the folic acid or folate, it is trapped into N5-methyl tetrahydrofolate, because from this N5-methyl tetrahydrofolate, methylene tetrahydrofolate cannot be formed, which is required for the formation of thymidine monophosphate, which is further required for DNA synthesis. So because of that, this process of TMP formation will be hampered, and in turn DNA synthesis will be hampered, and the DNA synthesis, this process is arrested in the S phase. And because of this arrest in cell division, there is accumulation of megaloblast in the bone marrow, because there is asynchrony between the nucleus maturation as well as cytosol maturation. The DNA synthesis is hampered, but RNA synthesis still continues, and that's why there is accumulation of megaloblast. And due to ineffective erythropoiesis, the RBCs are immature and hypersegmented neutrophils are seen in the megaloblastic anemia. Now let's see the biochemical basis of neurological symptoms seen in vitamin B12 deficiency. It is because of demyelination of neurons, which is responsible for its degeneration as well as dysfunction. So it is because of the decreased and defective fatty acid synthesis in myelin sheath. So myelination is affected in the cerebral cortex and posterior and lateral column of spinal cord. Here both sensory and motor tracts are affected, and that's why it is called as subacute combined, because both sensory and motor tracts are affected. The condition is called as subacute combined degeneration. Now we know that for the formation of S-adenosyl methionine, which is the active methyl donor, and this methylation is responsible for the myelination. So for formation of this S-adenosyl methionine, vitamin B12 is required, because homocysteine is converted to methionine with the help of this vitamin B12, and if it is deficient, then S-adenosyl methionine cannot be formed, which is required for the methylation, and this result in the decreased concentration of S-adenosyl methionine, and that's why membrane lipid homeostasis is altered, and this due to decreased SAM, the methylation of biogenic amines, proteins, phospholipid and neurotransmitter in the brain is reduced, and this is responsible for degeneration and dysfunction of the neurons, and this results in the neurological symptoms. Another reason for this neurological symptom is vitamin B12 deficiency, that is the deficiency of deoxyadenosyl cobalamin, which is required for the action of methylmalonyl CoA mutase, and here in this process succinyl CoA cannot be formed, and there is accumulation of methylmalonyl CoA as well as propionyl CoA. So increased concentration of propionyl CoA inhibits the acetyl-CoA carboxylase enzyme, which is required for fatty acid synthesis, and fatty acid synthesis is important for myelination of neurons. So due to impairment of fatty acid synthesis, myelin formation is impaired. Now this increased methylmalonyl CoA due to deficiency of deoxyadenosyl cobalamin, this is competitive inhibitor of malonyl CoA, and malonyl CoA is involved in the fatty acid synthesis. So in turn it impairs the myelin formation. Now this methylmalonyl CoA, which is increased due to vitamin B12 deficiency, It gets incorporated in place of malonyl-CoA in fatty acid synthesis, and that's why there is formation of abnormal branch chain fatty acid, and this abnormal branch chain fatty acid disrupts the neuronal membrane structure, and this is responsible for all the neurological symptoms which are seen in vitamin B12 deficiency. The assessment of vitamin B12 deficiency includes estimation of serum B12 level. The normal range is 150 to 350 picogram per dl. Holotranscobalamin, it is the metabolically active form of vitamin B12. So the estimation of this metabolically active form will give the actual status of vitamin B12 deficiency. Peripheral smear will show macrocytic hyperchromic anemia. Anisocytosis, poikilocytosis will be seen. Anisocytosis means there are red blood cells of different sizes. Poikilocytosis means there is increase in abnormal red blood cells of any shape, and hypersegmented neutrophils are also observed on peripheral smear. Bone marrow aspiration will show marked erythroid hyperplasia, which means that there is increased count of erythroid precursor cells. Methylmalonic acid level will be increased in both plasma as well as urine, and homocysteine level will be also increased in plasma as well as urine. Then there will be increased plasma indirect bilirubin. Pernicious anemia, which is due to the deficiency of intrinsic factor, and it is the autoimmune disease where antibodies are developed against the intrinsic factor, and that's why the absorption of vitamin B12 is hampered. It can be detected by detection of antibodies to the intrinsic factor, by detecting chlorhydria through gastric juice analysis, and there is absence of intrinsic factor in gastric juice. Another test is called as Schilling test, and this test is not used to evaluate the status of vitamin B12, but it is used to see the absorption of vitamin B12, and it shows the impaired absorption of B12. Now in this test, radioactive labeled vitamin B12 1 microgram is given orally, and simultaneously an intramuscular injection of unlabeled vitamin B12 is given, and in order to saturate with normal vitamin B12. So the radioactive vitamin B12 will not bind to the body tissues, and therefore in normal person the entire absorbed radioactivity will pass into the urine. However, in patients with pernicious anemia, less than 5% of radioactivity is detected in the urine, but nowadays this test is not done. Now the best method for assessment of vitamin B12 deficiency is estimation of holotranscobalamin as well as methylmalonic acid in plasma and urine. How the laboratory findings will help to evaluate the megaloblastic anemia due to folic acid deficiency or vitamin B12 deficiency? Let's see. In folic acid deficiency, the serum folate level will be decreased, but vitamin B12 will be normal. The methylmalonic acid level is normal and homocysteine level is increased. While in the megaloblastic anemia seen due to vitamin B12 deficiency, serum folate level is normal, but vitamin B12 level is low. Here the methylmalonic acid level is elevated and homocysteine level is also elevated. Vitamin B12 deficiency is treated, or megaloblastic anemia is treated by giving the supplements of both folic acid as well as vitamin B12. Vitamin B12 is available in oral as well as injectable form. Folic acid alone should not be given in the macrocytic anemia because it may aggravate the neurological manifestations of vitamin B12 deficiency. So both should be given, both folate as well as vitamin B12 should be given. So in this today's video we have seen the various sources, RDA and the biochemical functions, deficiency manifestations, laboratory diagnosis of vitamin B12 deficiency. I hope this video will be useful to you. Thank you for watching and happy learning.